Hi everyone. So today I'll give you a very brief overview of 8 years of the Human Connectome projects in 10 minutes or so. But hopefully I'll convince you that it's a very powerful method to use for investigating brain function. So I'll start with what's wrong with the traditional volumetric approaches that are commonly used. And the problem is that they heavily rely on cortical folds. And we know that from empirical findings that cortical folds are very variable across individuals, especially in higher association areas. I have this. Yeah, perfect. So they are very variable, especially in higher cognitive areas, and especially even between twins. So as you can see here, for example, between twin W and twin X, there's a difference in the sulci patterns across both of them. So for starters, this is not a reliable method to align brains together. Another problem is we traditionally align data to the MNI template, but the MNI template is actually bigger in size than the average brain volume of a subject by approximately 37%. So when you align it, you get a decrease in the variability across subject, but again, you just you artificially inflate the size of the brain. A third problem, for example, is the unconstrained spatial smoothing, which doesn't respect neuroanatomical features. So just by using something like 4mm smoothing, you can see that you've already started mixing signals across the gyri across the sylvian fissure. And when you start using something like 8mm, then you're already creating new anatomy and creating new gyri and sulci, and you're mixing signals from the white matter as well. So these are well-known problems, yet the community has persisted in using this approach because it's easy, right? The more difficult approach is to use surface-based methods, which is more accurate because it builds a geometric model of the cortex, which respects the neuroanatomy. The methods that you use to analyze data on the surface then force you to respect also all of these neuroanatomical features. And this has been demonstrated several times that alignment using surface-based approaches produces more sharp and better alignment across subjects than volumetric-based approaches. But still, they are not exactly perfect. So what can the ACP methods add to that? First thing is to respect brain anatomy, so acquire data informed by the brain anatomy. So this is a map of the cortical thickness across the whole brain. As you can see that the thinnest cortex is around 1.5mm. So to ensure that you have at least 2 voxels representing the thinnest piece of the cortex, the ACP recommends acquiring structural data at half of this resolution, so with voxels that are 0.8mm or less. And to acquire also functional MRI data with high resolution, then it's recommended to acquire voxels which are smaller than half of the mean thickness of the cortex, so less than 2.5mm. And you can clearly see the advantages of using something like 0.7mm T1 versus the standard 1mm acquired in finding the gray-white matter boundaries. Also, you can see the advantages of using a T2 scan to also capture and separate the gray matter signal from blood vessel signals and the CSF, which have very similar intensity if you're using just T1, but the T2 helps separate these two. An important tenet in the ACP approaches is to also do distortion correction, which fortunately is adopted a bit in the current field. To be as faithful as possible to the subject's physical space. So there are several types of distortion corrections. This is an example of what happens when you correct for the gradient distortion correction. To also correct for readout distortion corrections, the ACP recommends using spin echo field maps. Another important approach that the ACP uses is to acquire functional MRI data with very short TRs. When you look at this power spectra from resting state networks, you can see why. If you're using 2 seconds, a TR of 2 seconds, then the maximum frequency you can look at is something around 0.25 Hertz, and you're not already capturing a lot of the noisy physiological signals that contaminate fMRI. So if you have faster fMRI data, you can separate these signals together. Also, there's some evidence now that there are some neural-related board components even at 0.5 hertz. So if you're acquiring a lot fast TRs, you have a lot of data points, then you can use methods like ICA fix. So ICA will be better at separating these noisy components, and fix refers to an automatic classifier that classifies whether these components are a good signal or are a signal component or a noisy component. And when you analyze the data without cleaning using ICA fix, you can clearly see that it's a bit noisy. But when you apply ICA fix, then it's really much cleaner. And you can see here from the power spectra as well, it's improved the quality. It's removed the noisy components in the data. One of the most central tenets in the HCP approach is to use more reliable features related to cortical areas to align the brains together. So I've already mentioned that folding features are not that good because of their variability. So what we can use is something like myelin maps. Myelin maps. So if you take the ratio between a T1 weighted image and the T2 weighted image, you amplify the contrasts in these images that are related to the myelin content, and you can already see some very nice patterns. So for example, the somatomotor strip, the auditory cortex, the empty visual regions, they are all highly myelinated. So these are already very stable features that can be used to align these areas across subjects. And the higher association areas have also interesting gradients. You can also use resting state connectivity maps. So for each subject, you can extract several ICA components, and this can give you an idea about the topographic organization of these areas. So the approach to use these aerial feature maps is called multimodal surface matching. If you look at this specific task fMRI contrast, so this first row shows you the significant clusters if you just align your subjects based on folding patterns. But if you start relaxing your dependence on folding patterns, then you start to see a small frontal blob, significant cluster. If you start using myelin maps, then you start to see a bigger cluster. And if you start using resting state maps, then you get a much larger and more significant cluster. If you use all of these features together, then this approach is labeled MSM all. So you initialize the alignment using cortical folds, and then you use myelin maps and resting state networks, and obviously MSM all outperforms just folding-based patterns in delineating these functional areas and improving your final fMRI statistics. So armed with these high-quality multimodal data, it was time to then find some landmarks in the cortex. So what previous studies have done is that they've used one of these features only to divide the cortex into several regions. What the HCP approach has done is to avoid the biases of using a single modality and find conversions from different modalities. So for example, in this area, I'll just guide you through how they define one area. For example, area 55P is lightly myelinated than the surrounding ones. It's selectively activated during a language task. It has a unique connectivity profile to some temporal regions, not the dorsal visual regions, and it's thicker than the surrounding regions. So how can you draw lines? The common approach used in the literature is to have statistical thresholds, but statistical thresholds are not necessarily going to follow the biological boundaries between areas. A different approach is to use gradients, which is the first spatial derivative, or in other words, it highlights regions where there is a fast change in a small cortical area, which is more related to biological borders, and better than the statistical thresholds. So if you draw the lines across the hottest lines here, you will identify the biologically relevant borders. And in the end, they also related this area to old cytoarchitectonic and histological staining studies to have some sort of stability in and relation to the neuroanatomical literature. And that's how they defined, that's how they parcellated the whole cortex into 180 different regions. That's the now famous multimodal parcellation or Glasser parcellation. And it's 1.0 because this is obviously not the final say. But the ground truth is estimated to be around 200 areas. So to go further, because there are individual subject variabilities, they trained a classifier based on based on all of these multiple features, and this classifier was able to identify these areas in an independent set of subjects. So, for example, for the area 55B, this is a typical location for it. There are some subjects where the area is split, and the classifier is able to identify the split. And there are some subjects where the area is shifted from its typical location, and the classifier is able to identify that as well. So going back to comparing ASP methods with the current traditional volumetric approaches, these are three example areas: the probabilistic maps identified in each subject using the classifier I've just mentioned, and this is the overlap across them. And you can see here that on the surface, the reddish areas, the overlap is around 100%, actually, or even 90%. While in the while moving these parcels back to their volumetric to this subject's volumes, and then analyzing the data or registering the data using the current volumetric approaches, the nonlinear registration, for example, you can see that the overlap is good in areas like the motor cortex because the cortical folds are kind of stable. But once you start going into like higher association areas, you can see that the overlap is really poor. And this even becomes poorer when we smooth across the volume, because we have to for signal to noise ratio. But when you smooth across the surface, you can see that the overlaps are pretty much still consistent. So the second part of the talk was about how we use these methods to answer a question that our lab is interested in, which is identifying domain-general brain regions. Domain-general brain regions is a set of frontoparietal regions that is co-activated across many different types of tasks. And this is a famous study where showing the average across 7 tasks using 2 mm smoothing and unthresholded. We used HCP data to look at this in a more precise detail. So we used 3 of the HCP data, MSM all aligned, and only just 2 mm smoothing. And can you already see that this is providing a much clearer picture of this. So you can see that there are like different blobs here that are clearly delineated and separated from each other. And if you look at a flat map of the left hemisphere, you can see that it's really just like small islands in a big ocean that is identified. So it's similar, but each of these islands can be related to several cortical areas. So we have a much better detail at it. And to relate it to the parcellation, we are able to identify 10 core regions, which are the most strongly activated across these 3 tasks. So just 10 regions out of 180 regions. That's very precise. Then we collected some data here at CBU. Sneha and I did that. We took over the MRI building last year for around 3 months. We scanned for 200 hours, which was lots of fun. We basically collected 5 tasks. One of them was a visual NBAC task, and this is the group average from 37 subjects. And this one is the HCP NBAC task, and they are different in that the HCP is 2 back versus node back, and the ones we collected are 3 back versus 1 back. And I'll just leave you for a moment to appreciate how these 2 are similar, even though they are completely different scanners and different number of. The HCP is 450 subjects. We have just 37 subjects that we collected. So you can see that there are some striking similarities, like for example, this dorsal spread in the frontal lobe, and but there are also some interesting differences here. So the hotspot here in the frontal lobe is more ventral, while here it's more dorsal, which probably reflects the difference between the contrasts. And I'm only showing you this because the next figure is going to be questionable. So this is the two tasks, a visual and back task and an auditory and back task, again from the same CBU subjects on 2 different sessions. And I'm not going to tell you which one is visual and which one is auditory. You have to tell me. Any guesses?
Great question. They're both NBAC.
Yeah, they are both NBAC tasks. One is visual, one is auditory. Well, okay. This one is the auditory one, and what can give it away is the very faint auditory activation here. But this is so strikingly similar. Like John still doesn't believe that this is correct. He still thinks we screwed up in the analysis somehow, but it is true. You can see that they're not identical, but it is strikingly very similar. So what this tells you is that any shift, if we look at other tasks, any shift from these patterns of activation is meaningful and can be meaningfully interpreted as something that is probably cognitively different or at least physiologically different.
Is this a contrast of something?
Yes, sorry, that's 3 back versus 1 back.
Okay.
Yeah.
So would you expect major differences if you contrast, if you have, independently as to whether one is visual or auditory? Would you still expect differences? So one is visual, one is auditory. You have 3 minus 1.
Yeah.
Are you not left with memory component?
Yes, exactly. So that's the similarity, but there are some studies in the literature showing that visual and auditory and back activate different regions across the frontal cortex. But I'll come back to that in a bit.
I mean, can I just say something? I would expect if you did the exact same task twice with those subjects, it would look wildly more different from this. That's what's surprising.
Okay. Yeah. So just to wrap up, what are the data requirements for the ACP? Practically, you need 2 structures, T1 and T2 at 0.8 or less. That takes around 15 minutes, and then you need some functional data, ideally at 2 millimeter, and ideally the TR is less than 1 second. And you should acquire also feed maps, and single band reference is already acquired automatically. But how much resting state data or functional MRI data should you acquire for your MSM1 registration? That's still under investigation. But if you look at this data from the Midnight Scanning Club, you can see that the stability of network assignment actually starts to plateau around 30 minutes. So it's for now recommended to acquire at least 30 minutes of fMRI per subject. So in total, that means that you need 45 minutes just to do the HCP pre-processing. One last plug, which is if you don't have HCP style data, but you still want to relate it to the multimodal parcellation, what should you do? What you shouldn't do is to just move it directly from the MNI volume to the surface, because this is just full of errors. So all of these yellow patches at the top show you the vertices which are contaminated by white matter voxels, white matter signal. And these show you how the aerial borders here become very uncertain when you do this. The only correct approach is to reanalyze your subjects using surface-based approaches. There is already a pipeline called Siftify. It's Python-based. Whatever you have, you just have T1. If you have T1 and T2, or if you have functional MRI, it builds on the free surfer outputs, and you can then end up doing MSM sulc or MSM myelin or MSM mode. And this is the final slide, just to show you the difference between the current approaches in the peak area probabilities, so the maximum overlap across subjects. You can see here that it's a mean of around 50%. I mean, no wonder why fMRI is underpowered, really. But then if you just use MSM sulc, then that's already a major improvement. And if you use something like MSM-OL, then you have peak areal overlaps that are above 90% for most of the cortical areas. software requirements is pretty, everything is public, FreeSurfer, FSL, Workbench, and the HTTP pipelines are available from GitHub. And yes, thank you especially to the imaging team and the radiographers who had so much fun, of course, I'm sure, with us, and Jeff in helping us set up the pipelines on the imaging space. Thank you.